This series of videos they're all going to be about enzymes. And what is an enzyme? I've used the word before, but what you should know is that an enzyme is just a catalyst. And what is the word catalyst? Catalyst means something that will speed up a reaction. There are inorganic catalysts. We can speed up reaction using certain metals that will speed up a reaction. Hydrogenation will occur if you have a platinum or nickel catalyst. But in your body, all of the reactions need an enzyme. They get sped up, go faster, they actually would take forever without the enzyme. So your enzymes speed up reactions. They are organic catalysts. That means they are molecules that have carbons and hydrogens in them. And what are the typical enzymes? They are typically protein molecules. So remember when we were talking about functions of proteins, one of the functions was that they can behave, that you can have enzymes, or enzymes are proteins. The majority of enzymes are proteins. Okay, so we categorize enzymes into 6 different categories. A lot of times they name enzymes based on what they work on, what the reactant is that they work on, or they name the enzymes on what the reaction is, what type of reaction it is. But in your book, they've categorized it into 6 different categories. So we're going to learn those 6 categories, and I'll give you an example of what each one is. Okay, so let's start off with the first category, oxidoreductases. What do you think that word sounds like? Oxidoreductase. Doesn't it sound a bit like oxidation reduction? So yes, the first category of enzymes are the oxidoreductases. These are the enzymes that catalyze redox reactions or oxidation reduction. So these catalyze oxidation reduction reactions. In one of the videos, I told you guys an example of a redox reaction where you had an NAD involved. NAD got reduced and then the other reactant got oxidized. Well, a lot of the enzymes that have NAD, which is a coenzyme that we'll learn about in a little bit, they are catalyzed by enzymes that are called dehydrogenases. So the big category of dehydrogenase enzymes, and you'll be seeing them in glycolysis and in the Krebs cycle when we get into metabolism in the next series of videos. All those dehydrogenases are under this category of oxidoreductase. So I'm going to write the word dehydrogenase here. And if I ask you, what is an example of an enzyme that is in the oxidoreductase class, you would say any dehydrogenase enzyme. What do the oxidoreductase enzymes do? They catalyze oxidation reduction reactions. Okay? All right. So the second group are the transferases. Just from the word, transferase means it transfers something. Hold on 1 second here. All right, let me get that. They transfer something. So when I was doing my PhD research, I was working on an enzyme called tyrosine kinase. That's why tyrosine is my favorite amino acid. So this enzyme, this kinase, transferred a phosphate group onto tyrosine. Okay, so tyrosine kinase or any kinase would be an example of a transferase. So what does a transferase enzyme do? It catalyzes the transfer of something. I'm just going to put something. It could transfer a specific metal. It could transfer, in case of tyrosine kinase, a phosphate group. Just transfers something. All right. Doesn't sound very scientific, but that's what it does. All right. What's an example of a transferase? kinase, okay? My enzyme, tyrosine kinase, once it transferred a phosphate group onto tyrosine, that was an indication for us that these cells were becoming precancerous. If any of you are interested in all of my lovely research, I'm more than happy to tell you, maybe when I see you guys. All right. All right. What else? Hydrolase. Okay. Just from the word hydrolase, it sounds like hydrolysis. So all those enzymes that would hydrolyze an ester bond or hydrolyze a glycosidic bond or hydrolyze a peptide bond. So lipases, proteases, all those carbohydrates, lactase, amylase, sucrase, all those are under the category of hydrolase. So what is a hydrolase? It basically will break a bond using water. Okay, so the hydrolase enzymes catalyze hydrolysis reactions. So catalyze hydrolysis reactions. Hydrolysis reactions. RXN stands for reaction. I remember once when I was teaching, a student asked me, what does R times N mean? And I looked at it and I'm like, oh, this is just an abbreviation for reaction. It's not R times N. Anyway, so hydrolases hydrolyze ester bonds, glycosidic bonds, peptide bonds. An example of a hydrolase would be amylase. So the amylase enzyme, remember I told you about salivary amylase and pancreatic amylase, it will catalyze the hydrolysis of starch, amylose and amylopectin. Lactase is another example of a hydrolase. It will hydrolyze the lactose glycosidic bond. Remember the beta-1,4 glycosidic bond between galactose and glucose? That's lactase enzyme. It's under the category of hydrolase. Okay? The next one is lyase. And this one, I don't know if we've actually talked about a specific enzyme. So since we haven't, don't worry about a specific enzyme. But what you should know that a lyase basically is any enzyme that will dehydrate or hydrate. What does that mean? Dehydrate means removing water, and hydrate means adding water. Don't confuse that with the hydrolase, okay? This one is usually involving a double bond. That's what you tend to find with these lyases. So basically, if you're taking an alkene and adding an HOH and turning it into an alcohol, remember when we did that way back when we were doing organic? That type of enzyme would be under the lyase category. Okay. Isomerase, we didn't really talk about isomers, even though it's an important part of organic chemistry. Isomers are basically compounds that have the exact same molecular formula, but different structure. So glucose and fructose and galactose, those would be considered isomers. Remember, I told you all three of them are C6H12O6. So you've got enzymes in your body that will turn glucose into fructose. And so that enzyme would be an isomerase. All right. So put in your notes, isomerase will turn one isomer into another. Let me see if I need to push this down. You can see one isomer into another. And again, may as well put in your notes, what's an isomer? They have the same molecular formula, but different structure. There's an enzyme in glycolysis called phosphoglucoisomerase. So it's going to turn a glucose, a phosphorylated glucose, that means a glucose with a phosphate group on it, into a fructose with a phosphate. And it's just turning one isomer into the next, all right? Without that enzyme, you wouldn't be able to do glycolysis. So enzymes super duper important. All right, the last one is ligase. And this is, the ligase is usually are all of the synthetases. The synthetase enzymes means you're synthesizing a new bond. And usually you need ATP to do this. Okay, so a lot of synthetase enzymes, you need an ATP in order to get this done. So these are, the example would be synthetases, and you're creating a new bond. So you're putting two reactants together, and the only way you can put the two reactants together is you need a molecule of ATP for the energy to be transferred into those two bonds. Another transferase that you'll be seeing is a transaminase. I forgot to mention that. Transaminase. The transaminase enzymes, they help transfer an amine group from one amino acid into a keto acid to turn it into another amino acid. If you've had a nutrition class, you may have heard of essential amino acids and non-essential amino acids. Well, the essential amino acids are the ones you're getting from your diet, you have to get from your diet. And then when you consume those amino acids, your body can take the amine group from the essential amino acid, put it onto another molecule, and turn it into a non-essential amino acid. So these transaminase enzymes transfer an amine group. So if you don't want to remember the kinase that transfers a phosphate, you can remember the transaminase that transfers an amine group. Okay, so those are the 6 classes of enzymes. Know the 6 classes and know a little bit about each one, what each one does, and an example or whatever I've given you. Okay, so in the next video, we will continue on and we'll start using the terms in enzyme chemistry: active site, substrate, and coenzyme and cofactor. Yeah, we'll get into all of that in the next video. Okay.